Welcome back to What the Study Says. As a doctor, I've seen the heartbreak of cancer up close, but I have also seen what can only be described as miracles. And today, we're diving into a study that asks a question I honestly thought was pure science fiction. Is a universal cancer vaccine actually on the horizon? Let's break it down. So first, we have to tackle this huge puzzle that has been frustrating doctors for years. We've got these incredible drugs, immunotherapies, that can literally make tumors vanish. It's a miracle. But for most people, even with the exact same cancer, these drugs do almost nothing. So what on earth is going on?
Well, to get to the bottom of this, you have to understand how these drugs actually work. See, the problem isn't the drug itself. The problem is that many cancers have developed a brilliant disguise. They have an invisibility problem. Okay, so the most common of these drugs are called immune checkpoint inhibitors, or ICIs. The best way to picture this is to imagine your immune system's T cells as police cars, always patrolling your body for trouble. Well, cancer cells are master criminals who have figured out how to hit the brakes on those police cars, stopping them in their tracks. ICIs come in and just snip those brake lines, freeing up the T cells to go hunt down the bad guys. But here is the catch. Releasing the brakes on all the police cars doesn't do you any good if the criminals are wearing an invisibility cloak. A lot of tumors are what we call immunologically cold. They don't give off any signals that they're dangerous, so even with their brakes cut, the T cells just cruise right on by, totally oblivious to the massive threat sitting right there. But what if we could find a way to rip that invisibility cloak right off? A fascinating new study in Nature Biomedical Engineering suggests there might be a kind of tripwire we can use to do just that. And a secret to it is not what anyone expected. For the longest time, the conventional wisdom was that only cancers with a ton of genetic mistakes, what we call high tumor mutational burden, could be spotted by the immune system. The thinking was, the more mutations, the weirder the cancer looks, and the easier it is for T cells to spot. But this new study says, not so fast. The real key might be a much earlier, more basic kind of alarm signal. And that alarm signal is a protein called Type One Interferon, or IFN-I. The best way to think of it is as a cellular tripwire. When a cell gets damaged or infected, it pumps out IFN-I, which basically screams to the rest of the immune system, hey, get over here. Something is very, very wrong. It's the critical alarm that forces the invisible burglar, the tumor, out of the shadows so the police can finally see it. So just how crucial is this alarm? I mean, this data from the study is just staggering. In mice with cancer, the immunotherapy drug works beautifully. You can see that top line, their survival rate is great. But then, when the researchers blocked just that one IFN-I tripwire signal, look what happens. The therapy completely fails. That bottom line shows the survival rate just plummets. It was no better than doing nothing at all. This alarm signal, it turns out, is everything. Okay, so if this tripwire is the absolute key, how does it actually deactivate the tumor's invisibility cloak? The whole process is actually pretty fascinating. It all happens in this beautiful four-step process. First, the tripwire gets hit, and IFN-I is released. Second, that signal shoots up like a flare, calling in the immune system's first responders, cells like macrophages. Third, these first responders show up and basically rip the invisibility cloak clean off the tumor. And finally, with the tumor unmasked, the T cells, whose brakes have been cut by the immunotherapy drug, can finally see their target and go in for the kill. Now, this is where things get really wild. The scientists did this incredible experiment. They took T cells from a mouse where the therapy worked, and they transferred them into a different mouse with a tumor that was totally resistant to the therapy. And what happened? The resistant tumor started to shrink. Those T cells literally carried the how to fight this cancer instructions with them. But here is the absolute crucial point. That amazing T-cell transfer, it only worked if that initial alarm signal, the IFN-I, was present in the first mouse. If they blocked the alarm signal before taking out the T-cells, the transfer did nothing. It completely failed. This proves that IFN-I doesn't just trigger a one-time attack. It's what trains the T-cells and unlocks the entire powerful anti-cancer response. So this finding leads to an absolutely brilliant idea. If a cold tumor is really just a tumor that isn't setting off its own alarm, what if we could just install one for it?
And that is exactly what these researchers did. They created a universal RNA vaccine. Now, this is totally different from those personalized cancer vaccines you hear about that have to be custom made for each patient. This one is much, much simpler. It's just a generic piece of RNA, and its only job is to trick the body into thinking there's a threat, which in turn triggers that all-important IFN-I danger signal. So the vaccine is really just this tiny bubble of fat, a lipid nanoparticle, carrying a special type of RNA called uRNA. It's like a prepackaged, off-the-shelf alarm system that you can just inject, forcing any hidden tumor to suddenly become visible to the immune system's police force. And it turns out the type of RNA here is critical. The uRNA they used in this study was way, way better at setting off that IFN-I alarm than the modified RNA, or modRNA, that's found in some other vaccines. You can see it right here. For this job, you want the loudest possible alarm bell, and that's exactly what uRNA delivers. So the theory is fantastic, right? You just install a universal alarm to make invisible tumors visible. But did it actually work? Let's get to the results. The results in mice were honestly astounding. Those cold-resistant tumors suddenly became sensitive to immunotherapy. In fact, the vaccine was so powerful, it even had a strong effect all by itself. It dramatically improved survival and, this is huge, it taught the immune system to recognize the specific proteins on that particular cancer. Survivors were even protected when scientists tried to give them the same cancer again months later. That last point is probably the most incredible part of this whole thing. It's a process called epitope spreading. Think about it like this. The vaccine sounds a generic fire alarm that gets all the immune system's detectives to rush to the scene. But once they get there, they don't just put out the fire. They start investigating the arsonist, the tumor, and they learn its specific features. So the general alarm creates a highly specific and long-lasting attack. The immune system now has a mugshot of that cancer for good. Okay, so let's boil all this down. The success of our best cancer drugs might have less to do with how mutated a tumor is and everything to do with whether this IFN-I alarm gets tripped. This alarm is what unmasks tumors, and we can now trigger it ourselves with a simple RNA vaccine, potentially turning untreatable cold tumors into treatable hot ones. The authors of the study really put it perfectly. They suggest that what really matters for immunotherapy might not be finding the perfect specific target on a tumor, but just creating a loud, strong damage response somewhere nearby. Even an irrelevant alarm can be enough to get the immune system to focus its attention in the right direction. And this just leaves us with this provocative and incredibly hopeful final thought. Now, we're not there yet, but this research cracks open the door to a future where a single, universal, off-the-shelf vaccine could be the key that finally unlocks the power of immunotherapy for every single cancer patient. And that's all the time we have for this one. Thank you so much for joining us on What the Study Says as we broke down this absolutely incredible research. If you found this as fascinating as I did, please subscribe to the channel and share this with anyone you think might be interested. It is truly the best way to stay on top of the breakthroughs that are changing the future of medicine. We'll see you next time.